everyone, welcome back to my channel. Today I'll be doing part 3 Kingdom Monera Eubacteria. Part 1 and part 2 link will be given in the description box. Before we start this session, do not forget to like, comment, share and subscribe. Eubacteria are true bacteria and they lack nucleus and membrane-bound organelles like mitochondria, chloroplast, etc. Eubacteria are usually divided into 5 phylum. 1. Spirochetes, 2. Chlamydia, 3. Gram-positive bacteria, 4. Cyanobacteria and 5. Proteobacteria. The structural detail of the bacterial cell is been given in Kingdom Monera Part 1. The link has been given in the description box. Kindly go and check. Before going into the next topic of the eubacteria, we have to brush up the structure of the eubacteria. All bacterial cells are covered by a strong, rigid cell wall, which is made up of polysaccharide proteins and lipids. Cell membrane or plasma membrane, which is made up of lipoprotein, and it is thin, elastic, and also differentially or selectively permeable membrane. Flagella is fine, thread-like protoplasmic appendages, which helps the bacteria to swim in the liquid medium. Pili or fimbriae, which is besides the flagella, is a tiny small hair-like outgrowths present on the bacterial cell, and it helps in the sexual reproduction in bacteria. The inner part of the bacteria has chromosomes, which is usually single and circular. Along with chromosomes, there is presence of plasmids too, which are extra chromosomal genetic elements or DNA. A large number of ribosomes 70S type is present for the protein synthesis. Occurrence of the eubacteria. Eubacteria is found almost everywhere. So bacteria are ubiquitously found in the fresh water, maybe in the marine water, boiling water, ice water, sewage, etc. My dear students, there are approximately 4,000 species of bacteria in the world. So it is cosmopolitan. That means it can be found anywhere. The bacteria can be a friend, a bacteria can be an enemy to the human beings. Point number 2. If an average weight of the human is 70 kg, around 2.5 kg will be the bacteria, and here the bacteria will form a gut microflora which helps in the digestion and certain metabolic functions. Respiration in bacteria. Usually respiration occurs in plasma membrane of the bacteria. Respiration in bacteria takes place by aerobic respiration and anaerobic respiration. Under aerobic respiration, glucose is broken down into carbon dioxide and water using oxygen. This process is called as aerobic respiration. Anaerobic respiration, in the absence of oxygen and produce other molecules like nitrate. Reproduction of bacteria. Bacteria produced by both asexual and sexual methods. Asexual method is by primary fission and endospore formation. Moving on to asexual method. In this asexual method or reproduction, it takes place by primary fission. That means the bacteria, you can see on the screen, the bacteria is being divided both laterally and longitudinally. Next asexual method is endospore formation. Here the bacteria forms a very thick wall, resistant, dominant structure formed by the bacteria during unfavorable conditions only. Although the bacteria is not being differentiated by male or female sex organ, first method under sexual method is transformation. This was discovered by Frederick Griffith in 1928, and in this the genetic material is transferred from one bacterium to the another with the help of a conjugation tube. And conjugation was discovered by Lederberg and Tantum in the year 1946. Okay, here it is a transfer of a genetic material, as you can see on the screen, from one bacterium to the another with the help of the sex pili. The third method is transduction. This was discovered by Zinder and Lederberg in the year 1952. So in this process, the transfer of the genetic material is mainly by, or it occurs by, the bacteriophage, which you can see on the screen. Economic importance of bacteria. Bacteria decompose the dead bodies of animals and plants and convert them into simple organic compounds. It increases soil fertility. Hence, bacteria are called the world's best scavenger. 2. Fermentation. Many industrial products like butter, cheese, etc. are obtained by fermentation using bacteria. 3. Antibiotics. Many bacteria are used in the production of antibiotics. Example, streptomycin, erythromycin, etc. 4. Nitrogen fixation. Bacteria increases soil fertility by fixing the atmospheric nitrogen in the soil. Example, rhizobium, nitrobacter, etc. 5. Genetic engineering. Genetically engineered bacteria yield useful substances. For example, bacteria used here is E. coli. And the last one, 6. Mineral extraction. Mineral ores containing useful metals like gold, copper, iron, nickel, etc. can be extracted with the help of bacteria, which is called as bioleaching or microbial leaching.